Gram-positive aerobic bacteria. In gram-positive aerobic bacteria, there is two branches. One is cocci and other is bacilli. In the bacilli, further is differentiated on the basis of branching, and in the branching organism, Nocardia species, Streptomyces, Rhodococcus, and which is acid fast also, and Oresceva species and other similar species. While in non-branching catalase positive, which is Bacillus species, Bacillus species, Penibacillus species, and catalase negative, which is Listeria monocytogenes, Corynebacterium species and other similar species, and Elasopilithrix, Lactobacillus, Actinomyces, Corynebacterium species, Bifidobacterium, Gardnerella vaginalis, and other similar species. These are all catalase negative. Further, gram-positive aerobic bacteria are differentiated on the basis of morphology, that is cocci. In cocci, they are differentiated on the basis of catalase positive and catalase negative organism. In catalase positive organisms, Staphylococcus aureus and Micrococcus species, but in the catalase negative, we have Streptococcus species, in which the Enterococcus species, Abiotrophia species, Leucoconastic, Lactobacillus, Colabictella species, and Pediococcus, Aerococcus, Gemella, Helicoccus, and Alococcus, Otitis. We will focus on the Streptococcus family. In Streptococcus family, Leucoconastic may elongate to form the Coccobacilli structure, which differentiate, which is completely distinguishing property of Leucoconastic from the entire group of Streptococcus bacilli. Although cocci is the primary morphology of Leucoconastic, but they show little elongation to form the Coccobacilli type structure. Gemella species is easily decolorized and in the gram stain, and they show the resemblance like in Neisseria species. Pediococcus, Aerococcus, and Gemella, they are large cocci arranged in the tetrads, pairs, or individual cells. They do not show the typical Streptococcus chain morphology. Among the Streptococcus family, Abotrophia and Granulocytella is the two species. Both have extra requirement, and these both organisms do not grow at SBA and chocolate, and they require the pyridoxals. Pyridoxal discs are available commercially. Pyridoxal is basically vitamin B6. Vitamin B6 supplement for the medium is also available, and Staphylococcus aureus are also able to produce vitamin B6. So we streak Staphylococcus aureus with the suspected colony from the blood culture. Blood culture bottles commercially with the Castaneda medium have the nutritional requirement for the Abotrophia and Granulocytella. So it's easily grow on that medium. Further, in this group, one important organism in the group B Streptococci, that is Streptococcus aegylecti, that during the pregnancy are able to cause the diseases. So for the specialized media for this organism is toad havit broth with the antimicrobials like gentamicin, nalidixic acid, or other combination for the media supplement is colistin and nalidixic acid, which is also available in these antibiotics suppress the normal flora of vagina, and Streptococcus aegylecti grow. Another broth medium, which is LIM broth, is also useful in isolation of Streptococcus aegylecti. Differentiation of catalase negative gram positive coccidioid organism primarily found in chains. As we earlier explained that Pediococcus and Aerococcus and Gemella species, these are large cocci and arranged in tetrad pairs and individual cells. They are not found in the chain. So this is the another group of catalase negative gram positive cocci that are primarily arranged in the chains. The first organism is leucoconastic. If we see the table, in this table the first is the gram stain, and then hemolysis, cytochrome catalase, and then vancomycin resistance, LAP, which is leucine aminopeptidase, and PVR, pyrrolidoxal arylamidase. Both are available in the form of disc. And MRS broth for the detection of a gas, motility, and also bioluminescence, NaCl, and growth at 10 degrees centigrade and at 45 degrees centigrade. In this chart, the Enterococcus, which is vancomycin resistant, that Enterococcus is basically the differentiation of Enterococcus is very easy among the gram positive cocci, which is catalase negative, because the organism is non-motile and do not produce the gas. Rest of all the test of this organism is positive, because the organism can tolerate the salt water, and positive for bioluminescence, can grow in 10 degrees centigrade and also in 45 degrees centigrade. So for vancomycin resistance, the two species are the major species. One is leucoconastic and other is Vacilla. In leucoconastic, if you see, the leucoconastic is organism, if you remember, we discussed early that leucoconastic basically elongated to form the cocobacillary shape, while in Vacilla, which is large bacilli like colony, and both organisms produce gas in MRS broth. If we check the Vacilla, leucoconastic, and Pediococcus in this chart, you will find that the both Vacilla and leucoconastic produce the gas from the glucose. But Vacilla is positive for arginine, and leucoconastic is negative for arginine, while the Pediococcus do not produce the gas from glucose in MRS broth. Leucoconastic, Pediococcus, Lactococcus, Heliococcus, and Globicatella, and Tetra, Genococcus, and Streptococcus urinalis, Aerococcus viridans has shown the growth on bioluminescence and NaCl 6.5%. This is the reason these two tests no longer solely can be used to identify the enterococci. According to this chart, in streptococcal family, the Streptococcus bovis is quite a different organism, because they do not show the growth at 10 degrees centigrade, but can grow on 45 degrees centigrade. And this is a unique property of Streptococcus bovis, which distinguishes it from the entire group of Streptococcaceae. According to this chart, I just want to add that only Enterococcus family and Vagococcus have the ability to utilize the LAP and PVR both, but all other organisms have only ability to either utilize LAP or PVR. In this organism, in this chart, the Abotrophia and Granulicatella is a two organism in which both are positive for the LAP and PVR, but these both organisms required the cytalism around the Staphylococcus aureus, which is their media requirement, that is vitamin B6. If we see in Vacilla, the last organism in this chart, in the comments, the arginine is positive for this organism, while the leucoconastic is arginine negative. Enterococcus may produce a positive pseudocatalysis. This occurs when the Enterococcus faecalis strain grow on the blood containing medium are tested for the catalase production. Vagococcus fluvalis is negative for L. albinos and Raphanose, but Enterococcus gallinarum is positive for both. The most common isolates are positive, and Streptococcus pyogenes, pneumonies, and urinalis are PVR positive. In this task, the differentiation of the catalase negative gram-positive cochlear organism primarily found in the cluster are tetrads. Number one is Elococcus. Elococcus is vancomycin sensitive and positive for the LAP and PVR, but the organism is positive for NaCl 6.5. For it will take 2 to 7 days to give the results for NaCl broth. The organism is involved in the chronic otitis, but they cannot grow on anaerobically. Another important feature of Elococcus is that the organism do not grow in thioglycolate broth, so the gram stain must be done from the solid medium. D. pegarum grow poorly in thioglycolate broth. In this whole chart, only the Pediococcus organism is resistant to vancomycins. Elococcus, Feclema, Dolocigranulum, Pegarum, and Ignavironum, and Rothia, which is formerly called Stomatococcus. And these all are LAP and PVR positive. The rest of the organism are not positive for both LAP and PVR disc. Ignavigrarum rofi also need vitamin B requirement, and their growth can be enhanced with the Staphylococcus aureus cetilism. This chart is for the differentiation of a beta hemolytic streptococci. The species Streptococcus pyogenes, which have a large colony and Lancefield group A organism, that is only PVR positive organism in this group, and S. agenesis group, which is also belonging to Lancefield group A, and that is only positive for VP. S. agenesis group also belong to the group Lancefield grouping C and G and F and non-groupable. All of these S. agenesis have unique property that their VP is positive. In this entire chart, S. agilicti, which is belonging to the group B streptococci, have heparate and chem test positive. This is the unique feature of S. agilicti only in beta hemolytic streptococci. Streptococcus dia-agilicti, subspecies Ecomelis, is a large size cocci. They are grouped in Lancefield grouping group C and G, and the unique property of this organism, it is negative for PVR, VP, heparate, and chem test. The PVR and heparate or a chem test can be used to identify the group A, B streptococci respectively. However, use of 0.04 unit bacitracin disc is no longer recommended for the Streptococcus pyogene, because the group C and G streptococci are also susceptible to this agent, and S. pyogenes is the only species of beta hemolytic streptococci that will give a positive PVR reaction. Suspicious colony thought to be Streptococcus pneumoniae must be tested for either the bile solubility or susceptibility to optochin. In Streptococcus pneumoniae, the bile solubility test is a confirmatory test and is based on the ability of the bile salt to lyse Streptococcus pneumoniae. In the optochin test, which is the presumptive test, a filter paper and disc is impregnated with the optochin is placed on the blood agar and plate previously streaked with the suspected colony. and if it is susceptible, is considered as Streptococcus pneumoniae. A newly discovered organism, Streptococcus pseudonemonia, may interfere with the appropriate interpretation of the optogene disc test. The pseudonemonia are resistant to optogene, which is the zone is greater than or equal to 14 mm when they are incubated under the increased carbon dioxide. But pseudonemonia, Streptococcus pseudonemonia, are susceptible to optogene when they are incubated in immediate temperature. Therefore, the optogene disc test should be incubated under 5% carbon dioxide, and all tests should be confirmed by biosolubility test. This is one another chart which is useful to identify the Streptococcus family, which is alpha hemolytic. Mainly, if you see the optogene sensitive is Streptococcus pneumoniae, and optogene resistant is further tested by the VP acetone. And if a VP is positive, then arginine is used to test the organism. If arginine positive, and then PVR. If the PVR, arginine, and VP are positive but optogene resistant, the Streptococcus urinalis is the suspected organism. If the arginine and VP is positive and optogene resistant and PVR negative, that is est ageneous group, which is VP positive, as we explained previously. And if the VP is positive and optogene resistant, then we check for arginine is negative. Then if arginine is negative, then we go for the urease. If the urease is positive, this is Streptococcus salivarius group. If urease is negative, further it is tested by the help of mannitol. If mannitol is negative, urease is negative, arginine is negative, but only VP is positive and optogene resistant, that is considered Streptococcus bovis variant or Streptococcus salivarius. If the urease is negative and arginine is negative and mannitol is positive, and also the VP positive organism, we check for the beta gall. If the beta gall is positive, it is considered as Streptococcus mutant group, and if it is negative, it is suspected as Streptococcus bovis group. Further, in this VP negative group, we use the urease test, and if the urease is positive, then it is Streptococcus vestibulosus, and if it is negative, it is Streptococcus mitis group.